Good afternoon, and welcome back to AP Biology Semester 2. We're on the big idea, number 2, free energy. And of course, we are taking a tour of the cell in Chapter 6. Today, we are talking about the cytoskeleton and its network of fibers that organizes structures and activities in the cell. In the early days of electron microscopy, biologists thought that the organelles of a eukaryotic cell floated freely in the cytosol. But improvements in both light microscopy and electron microscopy have revealed the cytoskeleton, a network of fibers extending throughout the cytoplasm. The cytoskeleton, which plays a major role in organizing the structures and activities in the cell, is composed of 3 types of molecular structures: microtubules, microfilaments, and intermediate filaments. Okay, let's talk a little bit about the roles of the cytoskeleton and its support, motility, and regulation. Several types of cell motility also involve the cytoskeleton. When I say motility, I'm talking about the movement of the cell or the organism. The term cell motility encompasses both changes in location and more limited movements of parts of the cell. Cell motility generally requires the interaction of the cytoskeleton with motor proteins. Examples of such cell motility abound. Cytoskeletal elements and motor proteins work together with plasma membrane molecules to allow whole cells to move along fibers outside the cell. Motor proteins bring about the bending of cilia and flagella by gripping microtubules within those organelles and sliding them against each other. A similar mechanism involving microfilaments causes muscle cells to contract. Inside the cell, vesicles and other organelles often travel to their destinations along monorails provided by the cytoskeleton. For example, this is how vesicles containing neurotransmitter molecules migrate to the tips of axons, the long extensions of nerve cells that release these molecules as chemical signals to adjacent nerve cells. The vesicles that bud off from the ER travel to the Golgi along cytoskeletal tracts. The cytoskeleton also manipulates the plasma membrane in a way that forms food vacuoles or other phagocytic vesicles. And the streaming of cytoplasm that circulates materials within many large plant cells is yet another kind of cellular movement brought about by the cytoskeleton. All right, so the structures and functions of the cytoskeleton. There are 3 structures, as I mentioned before. The first one being the microtubules, which are tubulin polymers. Describing the microtubules are hollow tubes with wall. They're hollow tubes. Their walls consist of 13 columns of tubulin molecules. Their size in diameter is like 25 nanometers with 15 nanometer lumen. They have protein subunits, which are tubulin, a dimer consisting of alpha tubulin and beta tubulin. Their main functions are maintenance of cell shape, compression resisting girders, cell motility, as in cilia or flagella, chromosome movements in cell division, and organelle movements. The next structure is the microfilaments, which are also the actin filaments. The structure is 2 intertwined strands of actin, each a polymer, and actin subunits. Its size is about 7 nanometers in diameter. The protein subunit is actin. The main functions of the microfilaments is the maintenance of cell shape, tension-bearing elements, changes in cell shape, muscle contraction, cytoplasmic streaming, cell motility, as in pseudopodia, cell division, cleavage furrow formation. And the last structure of the cytoskeleton is the intermediate filament. Its structure is fibrous proteins supercoiled into thicker cables. The size is, it ranges from 8 to 12 nanometers in diameter. The protein subunits, one of several different proteins of the keratin family depending on cell type. The main functions: maintenance of cell shape, tension-bearing elements, anchorage of nucleus and certain other organelles, formation of nuclear lamina. Okay, so I hope you're getting this all in your notes because this is something that you will need to know in the very near future. Okay, now the components of the skeleton, of the cytoskeleton, and I had already mentioned before, and I'm going to mention again, about the microtubules. All eukaryotic cells have microtubules, hollow rods measuring about 25 nanometers in diameter and from 200 nanometers to 25 micrometers in length. The wall of the hollow tube is constructed from a globular protein called tubulin. Each tubulin protein is a dimer, a molecule made up of two subunits. A tubulin dimer consists of two slightly different polypeptides, alpha tubulin and beta tubulin. Microtubules grow in length by adding tubulin dimers. They can also be disassembled and their tubulin used to build microtubules elsewhere in the cell. Because of the architecture of the microtubule, its two ends are slightly different. One end can accumulate or release tubulin dimers at a much higher rate than the other, thus growing and shrinking significantly during cellular activities. This is called the plus end, not because it can only add tubulin proteins, but because it's the end where both on and off rates are much higher. Okay, let's talk about the centrosomes and the centrioles. In animal cells, microtubules grow out from a centrosome, a region that is often located near the nucleus and is considered a microtubule organizing center. These microtubules function as compression-resisting girders of the cytoskeleton. Within the centrosome are a pair of centrioles, each composed of 9 sets of triplet microtubules arranged in a ring. Before an animal cell divides, the centrioles replicate. Although centrosomes with centrioles may help organize microtubule assembly in animal cells, they are not essential for this function in all eukaryotes. Yeast cells and plant cells lack centrosomes with centrioles but have well-organized microtubules. Clearly, other microtubule organizing centers must play the role of centrosomes in these cells. All right, let's talk a little more about the cilia and the flagella. In eukaryotes, a specialized arrangement of microtubules is responsible for the beating of flagella, singular flagellum, and cilia, singular cilium. Microtubule containing extensions that project from some cells. Many unicellular eukaryotes are propelled through water by cilia or flagella that act as locomotive appendages, and the sperm of animals, algae, and some plants have flagella. When cilia or flagella extend from cells that are held in place as part of a tissue layer, they can move fluid over the surface of the tissue. For example, the ciliated lining of the trachea windpipe sweeps mucus containing trapped debris out of the lungs. In a woman's reproductive tract, the cilia lining the oviducts help move an egg toward the uterus. A cilium may also act as a signal receiving antenna for a cell. Cilia that have this function are generally non-motile, and there is only one per cell. In fact, in vertebrate animals, almost all cells seem to have such a cilium, which is called a primary cilium. Membrane proteins on this kind of cilium transmit molecular signals from the cell's environment to its interior, triggering signaling pathways that may lead to changes in the cell's activity. Cilia-based signaling appears to be crucial to brain function and to embryonic development. Though different in length, number per cell, and beating pattern, motile cilia and flagella share a common ultrastructure. Each has a core of microtubules sheathed in an extension of the plasma membrane. 9 doublets of microtubules, the members of each pair sharing part of their walls, are arranged in a ring. In the center of the ring are 2 single microtubules. This arrangement, referred to as the 9+2 pattern, is found in nearly all eukaryotic flagella and motile cilia. Non-motile primary cilia have a 9+0 pattern, lacking the central pair of microtubules. The microtubule assembly of a cilium or flagellum is anchored in the cell by a basal body, which is structurally very similar to a centriole. In fact, in many animals, including humans, the basal body of the fertilizing sperm's flagellum enters the egg and becomes a centriole. In flagella and motile cilia, flexible cross-linking proteins evenly spaced along the length of the cilium or flagellum connect the outer doublets to each other and to the 2 central microtubules. Each outer doublet also has pairs of protruding proteins spaced along its length and reaching toward the neighboring doublet. These are large motor proteins called dyneins, each composed of several polypeptides. Dyneins are responsible for the bending movements of the organelle. A dynein molecule performs a complex cycle of movements caused by changes in the shape of the protein, with ATP providing the energy for these changes. All right, now we're going to move on to microfilaments, or the actin filaments. Microfilaments are solid rods about 7 nanometers in diameter. They are also called actin filaments because they are built from molecules of actin, a globular protein. A microfilament is a twisted double chain of actin subunits. Besides occurring as linear filaments, microfilaments can form structural networks due to the presence of proteins that bind along the side of an actin filament and allow a new filament to extend as a branch. Microfilaments seem to be present in all eukaryotic cells. In contrast to the compression-resisting role of microtubules, the structural role of microfilaments in the cytoskeleton is to bear tension, pulling forces. A 3-dimensional network formed by microfilaments just inside the plasma membrane, or cortical microfilaments, helps support the cell's shape. This network gives the outer cytoplasmic layer of a cell, called the cortex, the semi-solid consistency of a gel, in contrast with the more fluid state of the interior cytoplasm. Microfilaments are well known for the role of in cell motility, particularly as part of the contractile apparatus of muscle cells. Thousands of actin filaments are arranged parallel to one another along the limb of a muscle cell, interdigitated with thicker filaments made of a protein called myosin. Like dynein, when it interacts with microtubules, myosin acts as a microfilament-based motor protein by means of projections that walk along the actin filaments. Contraction of the muscle cell results from the actin and myosin filaments sliding past one another in this way, shortening the cell. In other kinds of cells, actin filaments are associated with myosin in miniature and less elaborate versions of the arrangement in muscle cells. These actin-myosin aggregates are responsible for localized contractions of cells. For example, a contracting belt of microfilaments form a cleavage furrow that pinches a dividing animal cell into two daughter cells. Localized contraction brought about by actin and myosin also plays a role in amoeboid movement, in which a cell such as an amoeba crawls along a surface by extending and flowing into cellular extensions called pseudopodia, from the Greek pseudo, false, and pod, foot. Pseudopodia extend and contract through the reversible assembly of actin subunits into microfilaments and microfilaments into networks that convert cytoplasm from a sol to a gel. According to a widely accepted model, filaments near the cell's trailing end interact with myosin, causing contraction. Like squeezing on a toothpaste tube, this contraction forces the interior, more fluid cytoplasm into the pseudopodium, where the actin network has been weakened. The pseudopodium extends until the actin reassembles into a network. Amoebas are not the only cells that move by crawling, so do many cells in the animal body, including some white blood cells. My sinuses are killing me today. In plant cells, both actin, myosin interactions and sol gel transformations brought about by actin may be involved in cytoplasmic streaming, a circular flow of cytoplasm within cells. This movement, which is especially common in large plant cells, speeds the distribution of materials within the cell. Okay, we're going to talk about our last structure of the cytoskeleton, and then we're going to stop for today. Okay? Intermediate filaments. Intermediate filaments are named for their diameter, which at 8 to 12 nanometers is larger than the diameter of microfilaments, but smaller than that of microtubules. Specialized for bearing tension, like microfilaments, intermediate filaments are a diverse class of cytoskeletal elements. Each type is constructed from a different molecular subunit belonging to a family of proteins whose members include the keratins. Microtubules and microfilaments, in contrast, are consistent in diameter and composition in all eukaryotic cells. Intermediate filaments are more permanent fixtures of cells than are microfilaments and microtubules, which are often disassembled and reassembled in various parts of a cell. Even after cells die, intermediate filament networks often persist. For example, the outer layer of our skin consists of dead skin cells full of keratin proteins. Chemical treatments that remove microfilaments and microtubules from the cytoplasm of living cells leave a web of intermediate filaments that retains its original shape. Such experiments suggests that intermediate filaments are especially important in reinforcing the shape of a cell and fixing the position of certain organelles. For instance, the nucleus commonly sits within a cage made of intermediate filaments, fixed in location by branches of the filaments that extend into the cytoplasm. Other intermediate filaments make up the nuclear lamina that lines the interior of the nuclear envelope. In cases where the shape of the entire cell is correlated with function, intermediate filaments support that shape. A case in point is the long extensions, or the axons, of nerve cells that transmit impulses, which are strengthened by one class of intermediate filament. Thus, the various kinds of intermediate filaments may function as the framework of the entire cytoskeleton. And we are going to stop here for today. And the next time we will talk about the extracellular components and its connections between cells that help coordinate cellular activities. Until next time, ta-ta.